So can you hear me?
Yes, sir. Yes.
Yeah, so thank you very much for the introduction. I apologize not being there physically. And today I would like to talk to you about this intriguing structure that are the reducing body associated with different form of myopathies linked to a mutated gene that is FHL1. So the reducing body are not novel structure that has been described in 72 by Brook and Engel and Neville, sorry, associated with a congenital myopathy, where in the muscle biopsy of the patient were apparent these menadione alpha glycerophosphate dehydrogenase strongly reactive material that by ultrastructural study corresponding to closely packed, variably shaped osmiophilic particles. The story to identification of this entity has been long, and it was thanks to a study performed muscle biopsy with microlaser dissection of the reducing body followed by mass spec that the group of Kasten Bonneman identified that inside the structure there was an accumulation of this protein, FHL1, that was found in a muscle biopsy of patients with different clinical pictures, early onset myopathies or later onset myopathy with contracture. The FHL1 protein is encoded by this gene, that is FHL1, located in chromosome X, where the RNA cause the production of three different isoforms that are variably expressed in different tissues, including the muscle and the heart, specifically for the A and the C isoform, and the less quantitatively present, we found that FHL1B form in the muscle of the brain. And what is interesting that these isoform and this protein are characterized by different limb domains, and we will focus later on the limb 2 domain. What is FHL1 protein? So it's a protein that consists of these different limb domain that are composed by two distinct fingers in tandem, where we find highly conserved sequencing, and it exerts different interaction. The localization in the muscle tissue is at the membrane, the sarcolemma, but also it is found located at different areas of the sarcomere, notably the Z line, the I band, and the M line. And also there is an isoform that probably make a navette from the nucleus into the cytoplasm and vice versa. The function of these proteins are variable. There is a role that has been demonstrated in sarcomere assembly, and it interacts with this myosin binding protein C, that is a stabilizing of the thick filaments of myosin, but also it has been shown that it mediates hypertrophic biomechanical stress in response in the heart, and is also a regulator of the skeletal muscle mass. What are these FHL1 related myopathies? So we have different forms that has been described. The one with reducing body that interests us particularly, that it is associated with diffuse and severe respiratory insufficiency. We have form of rigid spine and form that specifically affect the axial muscle with this scapulo and peroneal phenotype with the distal involvement of the leg muscle. In this case, what is typical is that there is usually no cardiac involvement, while there are other forms that are not linked to the the reducing body in the skeletal muscle, where we can have an axial muscle hypertrophy, we can have cardiac disease with hypertrophic cardiomyopathy, or Emery-Dreifuss like phenotypes with this typical contracture at the elbows. The difference that has been hypothesized that in the forms associated with reducing body, we have an increase or normal or reduced FHL1, while constantly in this form that comprehend a cardiac disease, we have reduced FHL1 expression. Previously in our lab, we collected a cohort of 14 FHL1 mutated patients, and we decided to perform a comparative histochemical, immunohistochemical, and ultrastructural analysis in order to try to describe the sequential events that are leading to the reducing body formation. We studied 14 the muscle biopsy, and we clustered the biopsy into groups. Patients suffering from a reducing body myopathy, where the reducing body were found in the muscle biopsy and led to the diagnosis before the gene was identified, and another group where we presented phenotypes of Emery-Dreifuss-like or a hypertrophic cardiomyopathy. What we have done clinical and mutational analysis and deep morphological study with specific work on electron microscopy. Here, just to resume and synthesize the clinical features. So the first group that present invariably early or adolescent, early adulthood, I would say, onset disease, while the group two were presenting an adolescent or early onset either Emery-Dreifuss or hypertrophic cardiomyopathy. And when we analyzed the genetic variants, what we found is that the group of the reducing body myopathy all presented variants affecting the exon 5 of the FHL1 gene that is conserved in all the three isoforms, while for the cardiomyopathy we had a mutation in the exon 6, and for Emery-Dreifuss we have a more distal mutation in an exon 8 that do not affect all the isoforms. Here you have a representation of the effect. You can see here that in the reducing body group we have the LIMP2 domain that is affected in all the three isoforms, while for the Emery-Dreifuss or cardiomyopathy we have more distal variants that do not affect all the isoforms, while for example for hypertrophic cardiomyopathy we have only the A1A and B that are affected. But let's go into the muscle biopsy. Here we have sample images and representative histopathologic lesion. You can see that the muscle is very abnormal with the myopathic fissure, but the most important lesion is this accumulation of material that is pink with hematoxylin eosin, that is reddish with the fuxinophilic with the Gomori trichome, modified Gomori trichome. And then when we do this histoenzymatic reaction with the menadion with alpha-glycerophosphate, we found hyperoxidative areas that correspond to the protein aggregation. As confirmed by TPA staining, histoenzymatic reaction, we do not have myosin accumulating, and this is due to the body protein aggregation that really disrupt and invade the muscle fibers. Here you have other example. You can see these aggregates and the positive and intense alpha menadion alpha glycerophosphate reaction. We then wanted to characterize better these protein aggregates doing immunohistochemistry on serial section with different antibodies for the intermediate filaments. And when we analyzed the combination of an alpha-glycerophosphate menadion staining with desmin, we found that the desmin is found specifically around the reducing body, The same is for myothelin, that is really at the edge of this reducing body, and alpha-B crystalline was found specifically in fibers that were atrophic and also surrounding the reducing body. We then used monoclonal antibodies again against FHL1, and what it was evident that we had an accumulation of this protein inside the fiber, not only in correspondence of the reducing body, but also we had an overexpression in the group 1 of reducing body myopathy in the cytoplasm of the fiber. And here you have an immunofluorescence with a co-immunostaining of FHL1 antibodies with desmin, and also we found hyperreactive areas of HL1 that do not correspond to the desmin staining. But what was really intriguing and striking, that we found a peripheral halo of immunofluorescence of FHL1 around the myonuclei. These are the histopathological findings by light microscopy of the group 2, where we didn't find any reducing body. Here you have the menidion, and also when we did immunofluorescence for FHL1, what we found is that there was a kind of reduction or small, not very consistent accumulation, maybe only at the sarcolemma, subsarcolemma areas. But let's go into the electron microscopy studies. Here we found in areas of the sarcomere that were more preserved, and then we started observing lesions that seemed to arise from areas that are near that line or in the subsarcolemmal area that are associated with more filamentous material with subsequent degeneration of the sarcomere. As you can see here, you can have some disruption of the Z line with the Z line screening. The lesion then tended to become more patchy, more extended, and really dissociated the sarcomeric structure that you can see here. Is it becomes to be disrupted and dissociated, accumulating other autophagic material associated with the reducing body. When we analyzed that higher magnification, we found that these reducing body are characterized by coarse tubulofilamentous material oriented in different direction of the space, and you can see here that the Z-line streaming is surrounding this area where we found the reducing body. Another lesion that was frequently associated and found in all the sample of reducing body myopathy was the presence of these cytoplasmic bodies. These are protein aggregates that present a dense core with a clear halo of thin filaments surrounding, like a sun that is shining. And these were found not only surrounding the reducing body, as you can see here, but also intermingle with them. And you can see here in this patient where the muscle was completely disrupted and occupied by this granulofilamentous material that correspond to the reducing body. Then we focused on the nuclei, and a constant finding that we found was the presence of this osmiophilic granular material around the nuclei that corresponding to probably material that lately compose the reducing body. Here you have a completely formed reducing body that is surrounded by two nuclei, and one of them present this halo. One possible explanation could be that these isoforms that are affected by this gain of function variants may provoke an aggregation of the protein that cannot navigate anymore between the nuclei and the cytoplasmic region. We also found that this reducing in granulofilamentous materia, granular materia, was respecting some areas of the nuclear envelope, and sometimes we found in the nucleoles that the nucleoles were particularly dense in the nucleus of this muscle. And specifically, these granular material surrounding the nuclei corresponding to the immunofluorescence reactive material that we found with FHL1 antibody staining. Moreover, we could disclose a lot of different structures associated with the reducing body and the cytoplasmic body. Here you can have a fingerprint bodies that is composed by this material that is orientated like a fingerprint. We found also tubular aggregates associated and in proximity of the nuclei of the disrupted fiber. You can see here how the sarcometic structure is completely destroyed. And also we identified in the nucleus of one of the patient the presence of this nemaline body that is found in other forms of protein aggregate myopathy, like the nemaline myopathy, specifically the FHL1 related, but also the myopathy related, but also in form of myofibrillar myopathies as ZASP1. In contrast to the findings of the reducing body, we deeply analyzed the ultrastructure of the muscle sample with the other forms. The forms, I remember you, with the cardiomyopathy, and the other forms with the Emery Dreyfus-like phenotype. And what was really interesting that there was, in any case, any reducing body that we failed to identify. The only alteration that we found was this mild disruption with small sliding of the Z line and thinning of the myofibrils that we consider as non-specific. So with this deep characterization of the reducing body myopathy and other FHL1 related condition, what we found that the reducing body myopathy phenotype was severe and invariably affected the LIMP2 FHL1 domain. In contrast, the patient from groups 1 presented with a different phenotype, either contractile or hypertrophic cardiomyopathy, with a mutation that affected the distal part of the protein, and specifically the LIMP4 domain. There was a strict correlation between the genotype and the morphological phenotype, where the group 1 displayed this reducing body associated with a rich spectrum of different pathological changes. Notably, the cytoplasmic bodies were always associated with the reducing body myopathy sample, but also we found a large range of alteration, like fingerprint bodies, like sarcotubular aggregates, and even nemaline bodies inside the nucleus. Speculating about the dynamic of formation of these reducing bodies, we can think, based on our observation, that they arise from subsarcolemmal eye bands. And this was clearly demonstrated in the first images that I show you. And we can hypothesize that this aggregation later expand along different phenotypes and arrive surrounding the nucleus. Another hypothesis is that these proteins that are bulky cannot enter anymore, this isoform cannot enter anymore the nuclei, altering this interaction and this constant intertalk between the nuclei and the cytoplasm. So the two groups of conditions seems to be produced by different pathophysiological mechanisms. And the analysis of the consequence of the variants show that in the group 1 we seem to have a dominant negative effect of this truncated FHL1 protein that start an accrued protein precipitation, where we can see that the desmin and the other myofibrillar protein were more localized at the periphery of the reducing body that seems to be composed by FHL1 protein that is altered. In contrast, in the group 1, we can hypothesize that it is more an absence of the different FHL1 isoform that provoke an alteration of the skeletal muscle cell and provoke the phenotype with a particular phenotype that is cardiac in the form with hypertrophic cardiomyopathy, while the other present a muscular weakness but with a prominent contracture phenotype, like showing a Emery-Dreifuss-like disease. So this is all for my presentation. I will be happy to receive your question, and I would like to thank all my lab, and specifically Borna Ledas, who is the technician for electron microscopy that is doing an excellent job, and she took many of the images that I show you today. Thank you very much.